Ali, tell what is Fragile X syndrome?
Fragile X syndrome is the most common inherited form of intellectual disability. It usually presents as delays in reaching developmental milestones, like walking and talking. So kids with Fragile X usually will meet those milestones, but will meet those milestones slower than typically developing kids. It affects more boys than girls because it's an X-linked condition, so it's carried on the X chromosome. But girls can still get Fragile X syndrome. They just tend to present more variably. So, in addition to the delays in development, it causes intellectual disability, and often people with Fragile X syndrome will have features of autism as well.
You mentioned it's carried on the X chromosome. Let's take a step back and tell us about males and females and how we differ on the X's and the Y's.
Males have an X chromosome and a Y chromosome, and females have two X chromosomes. Fragile X syndrome is caused by this particular gene, which we all have, and it's called FMR1, which stands for Fragile X messenger ribonucleoprotein 1. And at the beginning of this gene, we all have this repeated sequence, and it goes CG, CGG, CG over and over again. So it's actually called a triplet repeat because it's these three letters that are repeated over and over again. And so most people have between about 6 and 54 copies of this code that's repeated over and over again. People with Fragile X syndrome have over 200 copies of this code. Now, the gene is really important in brain development, and it produces a particular protein, and that is what's really essential for brain development and functioning. So when people have over 200 copies of this repeated CGG sequence, for some reason that we don't fully understand, it actually switches the gene off. And so if the gene is switched off, then it can't produce that protein that's really important for brain development. And so that's why people with Fragile X syndrome have delays in their developmental milestones and have intellectual disability because that protein is not being produced.
Does it help to think about these genes and the letters like a piece of code?
Yeah, so another way of thinking about it might be that we all have a recipe book. So in the cells in our body, we have this genetic code and it's like a recipe book. Okay. And we have different sections of that recipe book. So we have salads and entrees and mains and desserts. And so those represent our chromosomes. So our DNA is packaged into 23 pairs of chromosomes. So different sections of that recipe book. And then within each section of that recipe book, we have different recipes. And we can think of those recipes as being like genes. Okay. So the genes provide the instructions to produce a particular recipe. If you're in the dessert section, you might be making a chocolate cake, right?
Mhm.
And we can have sometimes, and it's actually very common for us all to have variations in how those recipes are written. So sometimes you can have a variation in the recipe. It doesn't actually change your product. So you still get a really nice chocolate cake at the end of it,
It
right? You might have slightly more or less of a particular component or one component that's slightly different to the other, but it still ends up tasting the same. Like the way you might substitute, gosh, thinking about baking here. What would you
Mhm.
substitute? Say, for
Almond
example,
almond meal for something else.
exactly. Yeah, you might flour for almond meal, for example, and you still get a lovely chocolate cake at the end of it.
Yeah.
But there are other changes that you might find in the recipe where it actually changes the way that the product turns out. So your chocolate cake actually turns out differently. So if you are missing baking powder, your chocolate cake doesn't rise. And so just like that with our DNA, we can have changes in our DNA that actually can affect the functioning of that gene or the way that the product that the gene provides the instructions for is produced. And so sometimes those changes can be significant enough that the product that needs to be produced doesn't actually get produced or doesn't work the way that it's supposed to work.
If the mother is a carrier of Fragile X, does it matter whether the father is or isn't?
With Fragile X, because it's what we consider an X-linked condition, it's actually only the mother that needs to be the carrier in order to have a child with the condition. I was mentioning before this repeated sequence, CGG, CGG, over and over again. So Women who are carriers for Fragile X have between 55 and 200 copies of that repeated sequence. And what can happen is that can actually be unstable. And when it's passed on from mother to child, it can expand. So it can expand up over that 200 threshold. And so that's how a child can have Fragile X syndrome. Males who are carriers, on the other hand, because they have an X chromosome and a Y chromosome, they will pass their X chromosome with that genetic change onto all of their daughters. So their daughters will inherit it. But what's interesting about Fragile X syndrome is that repeated sequence, if they're a carrier, so them it's sitting somewhere between 55 and 200, that repeated sequence tends not to change in size when passed from father to daughter. Therefore, fathers who are carriers actually have a low chance of having daughters with Fragile X syndrome. But mothers who are carriers can have a daughter or a son with Fragile X syndrome, and they have a higher chance because their repeated sequence can expand when it's passed on.
When we have a child, if we're having a daughter, then that daughter will take one X from mum and one X from dad, because the daughter has
Correct.
to have two X's.
Yeah.
Mum's two X chromosomes, will they be identical in the gene that carries Fragile X?
So if mum's a carrier, typically one of her X chromosomes will have the usual number of repeats that most people have, and her other X chromosome will have a slightly larger number of repeats.
Okay,
What
so
we
there's a
sometimes,
chance you could get the X chromosome that is fine.
yes, yeah, yeah. So if she has that slightly larger number of repeats, it's what we call premutation. And that's the one that could expand to cause Fragile X syndrome in her offspring. So because she has two copies of the X chromosome, it could be that the copy with the usual number of repeats is passed on, or it could be that the copy with the premutation is passed on. So there's a 50-50 chance of passing that on for each pregnancy that she has.
So that's why you could be a carrier and have the lower number of repeats and you'd be fine. But when you have offspring, what you're saying is there's a chance that instead of passing on 55, that may change further. And now you might have 200 or more.
Yes, that's correct. And what gets more complicated with Fragile X syndrome is that if you're a woman and you have between 55 and 200 copies of that repeat, you've got that 50-50 chance of passing it on. But then there's also a chance of it expanding. So the larger the number of repeats within that range, the higher the chance of it expanding. So women who have a very small premutation have a very low chance of it expanding and having children with Fragile X, whereas women who have a much larger premutation have a higher chance of it expanding. So
So the
for
number, the
example,
total number of repeats will effectively go into a calculation that creates a risk score.
exactly. For example, say a woman who has 92 repeats has almost 100% chance that it will expand when it's passed on. Therefore, her chance of having children with Fragile X is actually 50%. Does that make
Okay.
sense?
Yeah, it does. As a genetic concierge or counselor, when you're looking at the test results, you're looking at how many repeats are on the female's two X chromosomes,
Correct.
and then making a calculation around risk profile.
Correct.
And that risk profile can be low. And at low, what is the one in how many chances? Or is that an unknown? It's just, we just say low. It's less than, what's 1 in 10, 1 in 20? What does a low look like in terms of a number?
So that's a really interesting question. So Fragile X syndrome is quite a complex condition to get your head around, and there's another element that plays a role here too. In women who have a small premutation, and when we do screening in the general population, we find that about 80% of women actually have a small premutation. So we'd call a small premutation between 55 and 70 repeats. Now, in that range, there's actually another factor that plays a role in the chance of having children with Fragile X. And that is that some people have something called an AGG interrupt. So remember, we have that repeated sequence going. Some people have an AGG in between those CGGs, and that can actually make the gene more stable. And those AGGs come up commonly in women who have that small premutation. So what we can actually do is do an additional test, AGG analysis, and that can help us to assess women's risk in that small range. If she
So
has a
female A
female,
could have the
yeah.
same number of repeats as female B, but female B could have the AG interrupt, which therefore makes her less
Yes.
likely to pass on that to the
Yes.
child. Right.
And if she has one or two AGG interrupts, there's a very low chance. With genetics, it's important to mention that we never say no chance. We always say low chance because there are always limitations in the testing technology, but there can also be other ways that people might actually be a carrier for a genetic condition. And we're talking about the CGG repeat, which is the most common way that someone can be a carrier, but there are other ways as well. Generally, we never use the term no chance, but certainly if a woman has a premutation in that small range and she has AGG interrupts, her chance of having children is very low. Once you get over 70, the AGG interrupts generally are less likely to be present and also play less role. So those larger repeat sizes over 70, the chance of having a child with Fragile X increases with repeat size up to about 90. And then once you hit 90, it's basically always going to expand. So as a genetic counselor, what we do, as you were saying before, is we look at the number of CGG repeats. If we need to test for AGG repeats, we do. And then we make an assessment of that chance of instability in the gene and the chance of having a child with Fragile X based on mum's result. And that's probably the most important thing for people to understand about Fragile X. It's actually understanding those CGG repeats and what that means. And in order to make that risk assessment, we look at the scientific literature, and that guides us as to what information we provide to that particular woman.
So all of this is a calculation really based on multiple factors to allow you to come up with a risk profile. And as you say, there's never a zero risk. Is there 100% chance when it comes to Fragile X? Is there almost a guarantee that it will happen? Or again, there's no such thing as 100%?
Yeah, so Fragile X is a really interesting condition because in general, the gene performs in a certain way and the repeat sequence has a certain risks associated with it. Uh, With Fragile X syndrome, there are always exceptions to that. So I don't think we would ever say to someone that they had a 100% chance of expansion to cause Fragile X in their offspring, but it could be a very high chance. What's interesting about this repeat sequence is there are some women who might have a premutation that's quite large and therefore high chance of having a child with Fragile X. But we have seen the repeat contract when it's passed on as well. So mostly it will expand, but occasionally it will contract and she'll actually have a child with a smaller number of repeats. We can never be completely sure how the Fragile X gene is going to behave, but there are certainly some very common trends. And so when we're talking to people, we'll explain all the different possibilities that may occur for them.
Over time, are we going to learn more about this and get more scientific on risk factors, do you think?
We are learning so much about Fragile X all the time. And absolutely, there is quite a group of researchers who have devoted their whole careers to specifically working on Fragile X syndrome. And there have been huge advances in our understanding because of the work of those people, and there will continue to be. So I think this is certainly a condition where we will learn a lot more and we will understand it a lot better over time.
With uh, some genetic conditions such as cystic fibrosis, there are now treatments coming onto market for sufferers of cystic fibrosis. Because this is more of a developmental concern, do we think that there would be potentially treatments in the future, or do we think that a child born with Fragile X syndrome will have neurodevelopmental problems that may not be able to be reversed?
I mean, I think we're in an era where we're learning so much about the genetic basis of these conditions and how how the genes work and their role and their function of the proteins that they produce. And I think ultimately we will find really significant treatments. At the moment, there are a whole lot of treatments and interventions that can help people with Fragile X, but there's no cure as such at the moment. I'm sure that with time, those treatments will improve. And there's also a lot of benefit that's been shown in early intervention for kids with Fragile X too.
And so let's say mum doesn't have this condition, but dad does, if when we do our 3-panel testing, only mum is tested, she doesn't have it, it doesn't have any of the other conditions, it's unlikely that dad to be will be tested. So how will we find out whether or not, if dad is a carrier, that is passed on to our daughter? Is they'll only be picked up when our daughter it gets genetically tested down the line?
Yeah. So because males who are carriers will have daughters who are carriers, males are unlikely to have children who have Fragile X syndrome. There's not a strong argument for screening males to find out if they're a carrier. And so really then it comes to, how would a daughter be picked up as a carrier? Hopefully when she comes to the point where she's thinking about starting a family, she's offered screening at that stage. If we're talking about people now, so we're talking about a male who's a carrier now and his daughter, we're talking maybe 25, 30 years down the track, she would hopefully be offered screening and she would be identified as a carrier for Fragile X at that point. So what we have to think about with screening is what we're trying to do is use our health resources in the best possible way that we can. So the aim of this carrier screening is to identify people who have an increased chance of having children with these conditions. And so males who are Fragile X carriers don't have an increased chance of having children with Fragile X. And so that's why we don't screen males for Fragile X.
So mum can pass on the X. So the son is XY, gets an X chromosome from mum that has the high levels of repeats.
Yeah.
But you could have a boy who has it or a boy who is a carrier but doesn't have the condition.
Correct. So if a female's a carrier, there are essentially 4 potential outcomes. So she can have a boy who inherits the genetic change and may or may not have Fragile X syndrome, depending on whether that repeated sequence has expanded in him. You can have a boy who inherits the other X chromosome, so hasn't inherited the premutation and won't have Fragile X syndrome. You can have a girl who does inherit the genetic change and it may or may not have expanded to cause Fragile X syndrome. So she may be a carrier or she may have Fragile X syndrome. And you can have a girl who inherited the other X chromosome, so therefore won't be a carrier or have Fragile X syndrome. If you take all of that together, there's a 50% chance for a woman who's a carrier passing that genetic change on. And then, as we said before, the chance of her having that child with Fragile X syndrome, depends on her particular repeat size.
How common is it when you have 2 X chromosomes for those chromosomes to be totally different? Intuitively, one might think that if I've got 2 X chromosomes, they're going to be identical.
Okay. So we have to remember that all of our pairs of chromosomes, we inherit one from mum and one from dad. Okay. And so if we go back to that kind of analogy of chromosomes are like sections of a recipe book, and then within that section of the recipe book, we've got a whole lot of recipes. A chromosome that we get from mum has a recipe for a chocolate cake on it.
Mm-hmm.
You can tell I like chocolate cake. And our chromosome that we get from dad also has a recipe for a chocolate cake on it. But it might be that there's some variation in the way that the recipe is written within the one that comes from mum and the one that comes from dad. So mum's chocolate cake recipe might have almond meal in it, and dad's chocolate cake recipe might have regular wheat flour in it, but it still makes chocolate cake. So we get the same genes from our parents for all of our chromosomes except the X and the Y, but they still have variations in them. If we think about like all of our chromosomes as being the one recipe book, it's like in the male's copy of their recipe book, they have one chapter at the end that's slightly different.
Okay, it's got the barbecue section.
Yeah. Yeah.
These are the types of analogies which I think you've got to tease out.
Yeah.
You know what I mean? For just the average idiot like me. Okay, get it. Because I don't think you'll ever totally get it.
Yeah. You don't have to fully get this. You just have to get the general idea of it.
Let's go through all scenarios. You've found out you're a Fragile X carrier.
Mm-hmm.
Is there a low, medium, high, or is it just a low and high? Well, you as a genetic counselor, what are you informing someone of their risk profile?
This is actually really important, and it depends on what laboratory you use for your testing. Some laboratories will just tell you a carrier for Fragile X and you've got this number of repeats. Other laboratories will say you're a carrier for Fragile X, you got this number of repeats. We've actually sent your sample off for some additional testing to really help us understand what that means. And then they'll come back and say, if you've got a small premutation, which is going to be most of the people who get these results, you'll say you've got this number of CGG repeats and you've got 2 AGG interrupts. So therefore, even though you have the premutation, the chance of having children with Fragile X. If you're in that larger range of the premutation, where you don't need to do the AGG testing, then you'll get a result that just comes back saying you've got an increased chance of having children with Fragile X and genetic counselling is recommended. And so then I personally think that anyone who gets a Fragile X carrier result should absolutely see a genetic counsellor, because it's really important to understand what that specific result means for you. And you need a genetic counsellor to help you interpret that. So interpreting a Fragile X carrier result is really outside of the remit of a GP or an obstetrician or fertility
And
specialist.
we're having this discussion as if I've just found out that I'm
Yeah,
a carrier, and we've been going for an hour, and there's probably another hour we could talk about this, right? To keep going from where
yeah,
we are now to where you would want me to walk out of this consultation or this discussion
yeah,
feeling empowered that I know what's going on and what my next best steps are.
yeah,
Now, that's just
yeah.
not going to happen in a GP clinic.
That's exactly why it's important to see a genetic counsellor. And that's where genetic counsellors fit in, is helping people to interpret their results.